Good morning, good afternoon, everyone. My name is Nawal Ouzren, and I'm the CEO of Sensorion. I'm very pleased to have the opportunity to share with you the Sensorion story today. I would like to start with some disclaimer. Sensorion is a public company, so every information I will be sharing today is public information. So Sensorion is a biotech that is specialized in the development of hearing loss therapeutics. And the company was created in 2009. And at the beginning, the focus was building a portfolio that could treat and prevent hearing loss. Last year, we actually completed a very important transformation with the signature of a strategic alliance with one of the top academic institutions driving the science of hearing. And this is Institut Pasteur. Institut Pasteur is behind the identification of most of the genes that are causing monogenic forms of deafness, but they also developed a portfolio of products that could hopefully restore hearing in monogenic forms of deafness. So let's start with some high-level biology. So let's look at the cartoon in the left. What you can see here in blue is the inner ear, which is a very small and very delicate organ. It's not very easy to access. If you look at the zoom in the middle of the picture, you can see that there are two important pieces, for the inner ear. You have the three-dimensional piece that you see at the top, which is responsible for the balance. And then you have the snail shape that you see at the bottom, which is actually called the cochlea, and that's responsible for the hearing. One point that is very unique about the inner ear and that I would like to highlight is that we are born with a limited numbers of sensory hair cells. We are born with 3,500 inner hair cells, which every single one of them is linked to a specific frequency. This is what gives us the ability to actually enjoy music, recognizes the voices of our parents or our beloved ones. And we are born also with 12,000 outer hair cells, which are like amplifiers. And unfortunately today, these cells do not regenerate. So once you lose the function, you lose the ability to hear. What today is a disadvantage is actually going to be a benefit for the gene therapy, because the expectation is that once you successfully transduce the inner hair cells or the outer hair cells, the target cells, the expectation is that the efficacy will be durable over time. Today, the scientists have identified that 119 genes exist that will cause monogenic forms of deafness. And what you can see on the left is that today, a lot of people are suffering from hearing loss. The expectation is that by 2050, around 900 million people are going to suffer from hearing loss. So it's a big untapped opportunity and a big unmet medical need. So the strategy of Sensorion is to build a portfolio that could be potential solutions for patients and for caregivers and doctors. We are using a gene therapy approach to restore auditory functions for people suffering from monogenic forms of deafness. We started our exclusive collaboration with Institut Pasteur with two preclinical programs. The first one is to restore hearing in autophagy deficiency, which is one of the most common forms of congenital deafness. And the next one is to restore hearing and the vestibular function for patients suffering from Usher type 1 syndrome. We're going to also develop a portfolio of products that can hopefully treat and prevent hearing loss, but this will be a small molecule approach. So here is the illustration of the pipeline of Sensorion. What's very interesting is that it's a balanced pipeline with clinical stage assets, but today I will focus on the gene therapy programs, which are at the bottom in the Restore section. So the first gene therapy program that is currently being developed with Institut Pasteur is completing in vivo proof of concept studies. We are ramping up activities to start the preclinical IND enabling studies. And the next one, which is focused on the Usher type 1 syndrome, is in the middle of the in vivo proof of concept studies. What's very important about the relationship with Institut Pasteur is that on top of the two programs that we decided to opt in and take as an option, Sensorion has a right of first negotiation for everything that will come up from Institut Pasteur in the field of the inner ear, in the field of gene therapy. And we have constituted a joint steering committee. And as you can see here, there are additional indications that we are interested in too, and that most probably we will be developing with Institut Pasteur. So now let's start digging into the details of the respective programs. So Usher type 1 syndrome is a very severe disease. It's a rare disease. The babies are born bilaterally deaf, and they are born also without vestibular function. So it means that they have issues holding their head. They are delayed in learning to crawl, in learning to walk. And by the age of 10 years old, they will unfortunately become blind. So these patients are completely deaf, they are blind, and unfortunately they have very serious balance issues. So it's a rare disease. Usher type 1 syndrome represents around 40% of all the cases of Usher syndromes. The prevalence of Usher syndrome today is not completely accurate, but publications are demonstrating that the prevalence in the 5 biggest European countries is between 13,000 and 55,000 patients. And for the US, it's between 13,000 and 56,000. And the focus of Sensorion with Institut Pasteur is specifically on the Usher 1G mutations. The next program that we are developing is focused on the autophagy deficiency. In this case, the babies are born bilaterally deaf, and they are not suffering from any form of syndrome. So that's the perfect proof of concept for gene therapy. The autophagy deficiency is actually one of the most prevalent causes for congenital hearing loss. The prevalence today is estimated around 20,000 cases in the US and in Europe, and the incidence is estimated around 1,100 newborns in the US and in Europe. Today, which is actually quite typical in rare disease where you have no standard of care or really poor standard of care, is that what you observe is a very delayed diagnosis. Today, there are no systematic identification of the gene mutation that are causing the congenital forms of deafness. So really, the gene therapy is potentially a life-changing opportunity for these patients. So now let's focus on the preclinical data that were generated by Institut Pasteur for Usher 1G. Let's start actually with the pictures in the middle. These are electronic microscopic pictures. And we start with the normal mice. What you can see here is that you can see the sensory hair cells. You have like this beautiful architecture of hair bundles that are really well organized, and you can see that the color shows the activity. What you can see also if you zoom is that there is a prolate shape, for this stereocilium, which is a hallmark of a functional tip link, which is very important. On the vestibular side, you can also see that you have the vestibular stereocilia that are also very well organized and really active. In the middle here, you can see that Institut Pasteur has developed an Usher 1G knockout mouse. And here you can see that unfortunately we lost the architecture of the hair bundles. We also lost, the typical prolate shape of the stereocilium, which means that we don't have this functionality of the tip link. It's even worse on the vestibular side. You can see that we don't even have the vestibular stereocilia. At the bottom, what you can see here is what happens once Institut Pasteur has treated with one single injection of an AV8 sense transgene, the Usher 1G knockout mouse. In this case, you can see that after one single injection, they've been successful in restoring the hair bundles architecture, and they've been also successful in restoring the prolate shape, which is the hallmark of a functional stereocilia and tip link. And on the vestibular side, that you can see they've been successful in restoring the vestibular stereocilia. What has been observed, from a histology perspective has been also measured from a functional perspective. And you can see on the right here, this is what happens, from a vestibular side. So Pasteur has been successful in restoring the vestibular function. In the middle here, you can see what happens, for an Usher knockout mice with the vestibular dysfunction. In this case, you can see that the mouse has this dysfunctional circling behavior. And once the knockout mouse has been treated, you can see that you don't see this circling dysfunctional behavior anymore. You see that the mouse displacement recording is actually very similar to the control mice. And what has been also observed from a functional perspective is that Institut Pasteur has been successful in restoring the hearing. And this is demonstrated by the electric current that was measured through the outer hair cells and the inner hair cells. You can see the comparison between the knockout mice and the treated mice for the bar chart. And the level they've been successful in restoring in terms of electric current is very comparable to the wild type. So that's very impressive. And these data were published in PNAS, which is a prestigious ENT journal. So let's move to the next program, which is autophagy deficiency. Here again is an illustration of the preclinical data that has been generated by Institut Pasteur. Let's start with the normal mice. You can see here in the inner hair cells the presence of the autophagy, which is this very important protein, important for the well-functioning of the auditory system. You see this beautiful green, which is representative of the activity. In the middle, you can see that Institut Pasteur has developed the autophagy knockout mice. And here it's very visible. You see no measurement and no activity of the autophagy. And then completely on the right, you see that Institut Pasteur has been successful in treating the autophagy knockout mice with one single injection of a dual AAV2 mutated with the autophagy transgene. And in this case, they've been successful in transducing in the target cells, and the autophagy has been fully restored, which is quite impressive. And what was observed from a histology perspective, again, has been measured from a functional perspective. And let's have a look at the auditory function. Here you can see the wild type. You can see the ability of the wild type mouse of hearing the click intensity at a different decibel level. You can see for the autophagy knockout mice in the middle, it's completely flat, so the mouse are completely deaf. And then after one single injection of the dual AAV2, you can see that the ability of hearing has been restored, and the waves are quite similar to the wild type. And that's also being illustrated by the ABA wave 1 latency here on the right. So what's also has been very impressive is that these data were quite durable over time. The effect has been measured up to 30 weeks, and they stopped at 30 weeks because they sacrificed the mouse. This data was also generated and published in PNAS. For the autophagy program, we are also working with a key stakeholder in the patient journey, which is Necker Hospital. Necker Hospital is the biggest pediatric hospital in Europe, but most importantly, it is the reference center, the European Reference Center for Monogenic Forms of Deafness. So they are quite well integrated within the ecosystem. And as we speak, we are working with them on a natural history, where Necker Hospital is now genotyping every single child that is being born deaf, and they are following then the natural development of the disease. What's also very important is that Necker Hospital is now engaging the dialogue proactively with the parents and the caregivers to inform them about the potential developments of gene therapy, so that hopefully once we will be in a position to start the clinical trials, we will have built a database of volunteers for that clinical trial. So I would like to conclude my presentation with some important milestones that you can expect from Sensorion in the next few months. Regarding the autophagy gene therapy program, we are in the final stage of securing capacity up to commercial development for the autophagy gene therapy program. We are going to communicate additional non-human primate data that are going to be very important to finalize the proof of concept study. Regarding the Usher type 1 syndrome, we are finalizing the proof of concept preclinical studies in the mouse. And we are ramping up activities to engage a dialogue with the regulatory authorities for our gene therapy programs. So with that, I would like to thank you for your attention.